Magic didn't work. There it goes. Boy, that's some slow thinking. All right. I'm going to do something tricky, which is to zoom this. Okay. So why am I doing this? This is obviously to get you thinking. You know, you can understand the ion gradients, the channels. You can draw a nice action potential. Hopefully, you're filling in, you know, the idea of the driving force, you know, for a particular ion. Okay, so that gives you an idea of, you know, where the direction and really the perhaps to some degree the extent or intensity of the current is. So you can now think about how the currents are flowing. That's the ion charges moving through the membrane. But there's things like this that make you think a little harder about it. And so that's what we're up to today. That's visible all the way in the back? You okay? All right. So this question was raised before in one of the lectures. It was just a little side. Okay. But it's an interesting question because it has ramifications. So could an action potential propagate backwards? And in this case, it's a condition where, let's just say you did it artificially. You stimulated at the synapse, at the presynaptic terminal, or at the distal, you know, at the end of the axon. You stimulated there strong enough so that it should be able to have depolarized the membrane enough to cause an action potential. I'm not necessarily looking for a vote, but what do you think? What do we say normally happens, right? There's this axon hillock, and then, you know, the neuron's basically a little computer. It gets all that excitatory and inhibitory input, and if that voltage change down at that axon hillock is high enough, then it will trigger an action potential, and it will, we say one of the first things is it's unidirectional. It goes down the axon and only goes in one direction. It's all or none, propagates, so therefore that signal is basically, you know, generated and, if you will, regenerated, you know, smoothly and consecutively as it goes along. But now we're going to stimulate at the nerve end. Okay? And we're going to stimulate enough that could cause the membrane to go over what should be threshold. So will nothing happen? Will an action potential go in the other direction? What do you think? Does it sound like a crazy question? Why are you asking this question? They go down to the synapse. You just told us signaling goes in one direction, you know? Yeah. This sounds crazy. Well, there might be a situation where this could happen.
So would it bounce off of it and come go the opposite direction then? If it's happening at the nerve?
Okay, so let's specify a little more clearly. That's an interesting. So one idea. I'm going to draw this into the side. Okay. Actually, let's just go ahead. We'll draw the whole axon. I'm not going to draw the cell body. Okay. And you get all the way down to the synapse, and the synapse is this little bit of a bulbous swelling for the presynaptic terminal, and there's vesicles and things in there. We're going to draw more of that today. And then there's a contact, let's say excitatory, so it's on a spine thing, you know, and then that goes on. So this would be the pre, this would be the post side, and then this, of course, is the axon. And normally, let's even say that's the axon hillock, so normally it would go like that. So now we're gonna stimulate, right here we're saying we're stimulating the nerve ending. So we were able to put an electrode in there, okay, so that allowed us to stimulate that. And there are membrane preparations, nerve preparations where, you know, the nerve ending is really big, and you could do this. Or there's another condition where that could happen. So the question is there directionality inherent in the arrangement of the sodium channels and potassium channels along the length of the axon so it ensures that it only goes in one direction? Or is it only going in one direction because it starts over here? This is the start point on the map, right? Well, you could flip a coin, couldn't you? Or you could think, okay.
It's because it starts over there.
So normally directionality is because it starts there. So to not belabor this, the exponential will go in the reverse direction perfectly fine. Because if you think about it, there's nothing about directionality in terms of the sodium channels. They're all, so this is unmyelinated, okay, in this case, it doesn't matter. Sodium channels are all along the length of the axon. Potassium channels are all along the length. They're not coded in any way. The reason there's directionality is sequence. So at this start point, you raise the stimulation raises the membrane potential just above threshold, right? Now you're getting a little more sodium coming in, the potassium going out. Remember, that's how we say is when you get the trigger, the threshold is when those currents are the same. So you get one more sodium coming in, as it were, and it takes off. Remember, it's timing. Sodium rushes in, okay, potassium follows. It's a timing thing. It's all timing that dictates directionality. So there's no problem with it going in the reverse direction because you stimulate here, sodium starts to rush in, gets past threshold, it's going to spread the potential. And there's no directionality in spreading that potential. Normally here, with a sodium channel, right? Sodium comes in, we're just going to leave it at that. Remember, it spreads that current in this direction. Well, it actually can spread the current in the back direction as well, all right? There's nothing about the axoplasm, that is the interior of the axon, that says it only goes one way versus the other. It's just when it goes backwards, behind that leading edge of the axopotential, what's happening with that membrane? It's refractory, so it won't respond to that. So when you stimulate current here, okay, it spreads this way, and you generate an axopotential going in the wrong direction. Okay, now we're going to get tricky. We're going to stimulate here in the middle. Okay, what will happen there? Both directions. In this case, you're starting with a resting state, a naive state, okay? So the current is going to go in both directions. You will get axopotentials in both directions. So directionality is because it starts in the cell body. The dendrites do there's a computation, if you will, again, in terms of pluses and minuses, right? You have excitation on one side, inhibition on the other side. So whatever that net effect is, if it gets above threshold, and then it's all time, right? Now, is this just a simple exercise, or is there a pathological condition where this can happen? Now think about a nerve anywhere in the body. Well, if there was a penetrating wound, right, into tissue, and it cut this membrane, it chopped into the axon, what would happen? So if that happened right here, okay, so now you have something that, literally chopped a hole here, right? Sodium's going to go in, right? Because now, this isn't a channel. There's still the gradient, right? It's going to collapse pretty quickly, but there's going to be a quick voltage change, right? It may actually, right in that spot, go to zero eventually because everything will equilibrate. But remember, zero is way above threshold, right? So initially, you could get a burst of axopotentials going the wrong way from some penetrating wound or damage, whatever way that happened, in the wrong direction. And it probably won't last for a long time, right? But it may be enough to disrupt signaling, okay? Then you could have the other situation where you had, damage to the middle of the axon, so they're going to be going in both directions. But now you have an axopotential going to the synapse, and this is completely unlinked to signaling, right? So you're going to have not only a burst of axopotentials going in both directions, but they're going to stimulate that synapse. Or in this case, if it's a whole bundle of nerve fibers. So anatomically, we usually talk about a nerve as actually a bundle of nerve fibers. So you talk about the sciatic nerve. It's full of nerve fibers there. So sometimes there's confusion whether this is just a nerve or anatomically call a nerve as a bundle. Either way, more often than not, a whole mess of them may get damaged. Okay. So I think it reinforces the idea that directionality is where it starts and timing. Okay. Now, let's talk about something else that's a little tricky. The action potential rises very rapidly. In this case, we're going to have a condition which is idealized, where remember the action potential goes between the equilibrium potential for potassium, a little above that, up to the equilibrium potential for sodium, right? Normally it never gets there, but in this case, we say it's getting up there. So rises quickly to that point before sodium channel inactivation occurs. What happens to, so now let's draw the action potential. Okay, and we'll say that that's right at E and A. And we answered this question actually in the last lecture. So what happens to the sodium current? So it goes all the way up there. And let's just say in this particular axon, this particular condition, maybe it's actually even a little flat up here, right? A little hint for the next question. So for a moment up there, it's right at the equilibrium potential for sodium, and inactivation didn't happen a little behind that. What happened? Yes?
The channel may still stay open, even though it's not inactivated, but not as much ion absorbed.
Right. Not as much, or?
None at all.
Zero, right? Remember? No net ions flow at the equilibrium potential. So the answer is yes. There'll be little ion flow. Actually, there'll be no ion flow because you're at the equilibrium potential for sodium. So the other reason that the voltage stops as you approach the equilibrium, so let's say you're a little bit below there, okay? Then it's still slowing down. Remember, driving force depends on how far away you are from the equilibrium potential. So let's say you didn't get all the way up there. You're here, and maybe even here. It's slowing down, and then it will stop, right? Because no net flow of the equilibrium potential. So it stops the action at the top of the action potential if it goes to equilibrium potential for sodium because no ion flow. No ion flow, no more change in voltage. It's just going to stop there, okay? It's actually the other way around. So the inactivation is really starting just at the top end of the action potential. And so the current is slowing way down, and then the ion channel is shut. And that's the case where you don't reach the equilibrium potential for sodium, but you get a perfectly sharp action potential, okay? All right, now we're getting more tricky. Did I go too far? If the sodium channels do not inactivate right after the peak, or normally even a little before, so no, a little before or even right after, but they take quite a while. How long is the typical duration of an action potential? Do you remember? Pretty fast, right? Is it up there? Oh, hell. Okay. All right, so how long is it? Okay. So about a millisecond, right? So now the inactivation is delayed for 2 seconds. So in the lifetime of physiology or neurophysiology, that's huge, right? That's 2,000 milliseconds. Okay? What's the action potential going to look like? So we're going to start here. We're going to go up, right? Because we've activated by stimulating. And I should have put a little bump in here, right? So a little bump, starting to go up. Will it go up perfectly fine? So no inactivation in this case, all right? No inactivation until much later. Where's it going to go? In this case, it's going to get much closer to the equilibrium potential for sodium, right? Because remember, said inactivation is already even starting a little bit. But also remember the potassium conductance is happening. So we're just going to say it's getting close to that. Okay, so now we're there. They didn't, and I'm just going to say that's again at the equilibrium potential for sodium. That may still not happen because of the potassium, but that's a little hint for the next question. So what's the action potential going to look like? What is driving down the action potential? So potassium's going to drive it down, correct. Okay. But now the sodium conductance is still really high, right? Didn't activate. Not a strong driving force, right? So right at the peak, the current for sodium should stop. But what happens with the delay? The potassium starts to go out. Its conductance has gone way up, right? So this, what the underlying assumption here is your conductance for sodium, as I said before, those channels are open, all right, but now they're not inactivating. They're taking a while to inactivate. Where will the next phase go? We have a little hint, remember? This is coming down to the equilibrium potential for potassium, right? Remember, very strong conductance or current for potassium, but we still have a very strong current for sodium. So now you have both. Joy.
Would it still go down but a lot slower?
So it may go down, and it may go down slower, okay. So part of the downward phase is not just the potassium current, right? It is the fact that the sodium channels are out of the picture. They've been inactivated. So the potassium going out, trying to bring that voltage down, in this case, is unopposed when there's sodium activation, right? Now it's opposed. So it'll affect the rate of the drop, yes. That's true. Because they're competing. Where's it going to go? And now you have, let's just say for purposes of discussion, but you've seen this, you have a very strong current, or conductance that is, for sodium, and you have a very strong current or conductance for potassium, depending on the position of the voltage. Okay, so starting to come down. That's true, it will start to come down. All right. Where's it going to go? Remember, the exponential goes between the equilibrium potential for potassium and sodium. All right. Now you have both the sodium and potassium working. All right. Remember, the reason why for the up phase is that along here, the sodium is overwhelming the potassium. You don't even worry about the potassium. But now it's coming down. Now they're both really strong. Let's say they're equally strong. I think we had so.
Yeah. So yeah, as you go down, the potassium's going like slower. So it's gonna just like flatten out a bit.
Very good. Was that your vote?
Yeah.
Yeah. It will go to some intermediate value. And then this is 2 seconds. So it's going to go and go and go and go and go and go. Okay, I'm going to put a little hatch in here. All right. And then eventually inactivates, right? So 2 seconds to do that. So then what will happen? It will then go back, go down, and you'll have a little undershoot. Okay. Well, this is really absurd, but there are cases where inactivation in certain nerve systems, neurons, you know, the generating system, does have quite a lag, is slow, all right? And so, more realistically, there are places where you'll see an exponential that looks something like this. Maybe a little bit more of an undershoot. Because of the scaling here, this should go on and on and on and on, right? So everybody clear on this? You have sodium going in, potassium going out. Normally at the top of the exponential, you take away the sodium. Potassium dominates, drives it all the way down to the equilibrium potential for potassium. Right? Now they're competing, and we're saying they're equivalent, right? So it's going to fall to an intermediate phase. And you do see that in certain neurons, particularly sensory neurons, for some reason, do this. In the sensory nerves going into the spinal cord. They have this long plateau in their exponential. Now you see a long plateau also in the cardiac exponential, but there's a different reason for that. But it's still because sodium is rushing in. Okay. So this is actually seen. That's a type of exponential you run into. Okay, let's move ahead so we can get to the main subject today. All right, I'm not going to spend as much time. Okay, if there's no increase in the conductance for potassium at any time during the exponential, what would happen to the following phase of the exponential? We'll start again. We're going to go up. Okay, we get sodium channel inactivation, okay, in this case, but we didn't get that delayed increase in potassium conductance. Remember, it's a balance of power. So he said that that resting potassium conductance that dictates the membrane potential, it's still there. It's just so small compared to that huge potassium conductance increase you get in the action potential. So now we take away that huge increase in potassium. So that little one's still there. Sodium channels have been activated at the top of the action potential. What will the action potential look like? So a balance of power. Now we have remaining a small, kind of piddly, but stable rest conductance for potassium. Remember, he said that that conductance for potassium is independent, that channel is independent of voltage. So it's still going to be working at the top of the action potential. But normally it's just swamped out. Okay, what's going to happen? Well, start with first principles. That, you've gotten rid of sodium, starts to look like your picture for the resting potential, doesn't it? This is the conductance that dictates the resting potential, so
The rate at which the voltage decreases will be less. Like it'll.
so the rate of the falling phase will be less.
Yeah.
Yes, actually it'll be
Would it be possible for
hugely
the
slow.
sodium channels to like activate again before it reaches?
Well, that's an interesting question. Now you're getting very tricky. So the question was, could the sodium channels reactivate? But remember, when they reactivate, they're still very much dependent on a fast depolarization. We're going in the wrong direction. So yes, they're going to, inactivation will start to be removed, but the channels will be closed. Okay, because they're going to, as inactivation is lost, the channels close. Okay, but it's an interesting question about timing of inactivation. So, and again, you'll see action potentials that are very, very slow to come back down. So they're real slow because you have this small movement of potassium, okay, and we're just going to ignore the inactivation, even though that inactivation is starting to be removed. Okay, those channels are basically closing as you go down. So we forget that. It's just taking a long time because that potassium conductance is so weak. But it will do its job. Why? Because the voltage will go, because this is a battery from the ion gradients, doesn't matter how slow it is, it will eventually find its way down toward the equilibrium potential for potassium. Now at this stage, probably won't make it there because this is so slow. Remember, there's that leak current, so it probably really won't have much of an undershoot. This is more likely the case. Okay. Is everybody clear on that? So the reason why that falling phase of the action potential is so fast, sodium channel inactivation, real big conductance for potassium. Okay, so it just slams it down. Now you have a little one, it'll still get there. It'll just take its sweet time. Okay, the last two will be quick, and I'll go with the first one for one last little tidbit. So what key experiment was used to determine, in this case, a potent neurotoxin to demonstrate that it's the influx of calcium that is the basis of the rising potential. So this is kind of an enforced recall question. Nobody's looking it up? Remember I said you can use all your resources. You could check with your neighbor. Well, let me tell you, toxin's in the name. Tetrodotoxin. Now the last one I don't want to spend a lot of time on. We talked about individual channel only opens for a short time. So how do the sodium channels come together to drive the action potential? A similar deal with potassium. So there's something here about inactivation. But so in the rising phase, what's really happening? Because the action potential, even the front end, is many, many hundreds of microseconds. So, but these are short openings, open, close, open, close. So what do we say? How does it come together to drive the rising phase? What's really happening when you get a voltage change? So to put it as an either or, obviously this implies then that the channels don't all open till the top of the action potential and all close, right? It's that takes too long. That's the underlying condition, or really presumption here, right? They're opening, closing, opening, closing, opening, closing. So remember we talked about it, what's really happening is across the population of sodium channels, right? So during that rising phase, with that drive of voltage you get above threshold, okay, more and more and more of the channels are opening, okay, and then they stop. You go back and look at that if you want in the text and chew on that a little bit. The other thing I like to draw here is, what do you think this, when you look at the axon, and now we're going to talk about the normal direction, but we could do it either way. And so the ax potential is going down this way, right, from left to right. What does the ax potential look like if you were at a fixed point and you watched it go by? The rising phase is at the leading edge, isn't it? So does the ax potential look like how we draw it? That's a time sequence, not a directional sequence. The ax potential will look like this. It will look backward, look reversed, right? Now are you completely confused? So you got up to this point, you thought, I'm following some of this. Why is it backwards? Gave you the answer, right? The leading, as the exponential is going down, right here, this is the current flow. This is the sodium influx here, right? This one, sodium. Behind is potassium, right? So if you sat on the axon and you watched the exponential go by, it would look reversed from what we normally show it, because we're showing the sequence, right? Now we're showing position. Again, to reinforce that, of course, sodium's going in here. So it should look like, to blow it up, it should look like that. Okay? Was that just 1 point too many? All right. But remember, again, first principles, as you're going down the axon, current is flowing down. There are the channels that are responding, right, that show the current, and we're really showing sort of a summary here, that show the biggest current are the sodium channels. So they're going to be the front end of the ax potential, right? So it will rise, and then it will fall behind because potassium's going out. So if you go through the sequence, you realize you have to draw it backwards. Actually, this really should be frontwards. We draw it backwards when we do it by time. Okay? Need to chew on. Okay. That said, work through these to help you think. There's other questions that I'll certainly pose during the review and as we go along. Does anybody have any questions now? Yes?
It's kind of obscure, might be too crazy a topic to start, but what's going on with ghost limbs? Like when people feel an itch from an amputated limb?
That's a good question. So, where's that signal coming from? So we're going to get into that when we talk about sensory systems in section 3, so we are a little ahead of ourselves, but that's okay. So remember, signals are transmitted, right? And we're going to talk today about synapses, right? So it turns out that there is, a synapse interposed between your limbs, right, and going up to the brain. We're going to learn about neuroanatomy as well. So because of that, all right, there's a way station, right? So normally that synapse would be responding to signaling coming down that axon. Now, it turns out that when you have either chronic pain or very severe signaling, that the cells in the spinal cord start working on their own. They start firing even though they get no input. And we don't completely understand why that happens. There's something to do with itch or pain and how it is funneled the sensory information. But you're right, you have no axons that are firing. Because remember, if we cut that axon, it'll fire for a while, okay? But then it's going to stop because you collapse the grating. But it's going to fire and fire really bad for a long time. So it turns out there's a crazy adaptation in the spinal cord where those cells just keep firing with no input. And so that's why people have either amputation, it turns out less so than with certain types of cancers that are causing really extreme pressure and tissue damage, say in a limb. Then you'll have this chronic pain. And it's actually, in most cases, very difficult to relieve. So now that signaling is uncontrolled, you can't intervene. So that's all part, in the end, our perception depends really on those signaling elements, you know, the patterns, whatever, the frequency, coming through all these stages. So what's happened is you've lost stage 1, but stage 2 is just taking a mind to do it itself. And this is a fundamental problem with that kind of neuropathic pain, we call it, or with certain types of chronic pain. And it can be very, very difficult to treat because it's completely disconnected from normal triggers for that sensation. So that's an interesting point, which is a nice segue for us to get into the synapse. Okay. All right, give me a sec. This one. All right. There we go. Okay. I know you can't see this all that well, but hopefully you can see it on your own handout. So now we're moving down. Yes?
Are you going to post it on Alima later?
It should have been posted. I could do that right now. Let me double check. Yeah, that should have been done. Sorry.
Am I just missing?
I
Sorry.
had a little trouble. Well, no, it'll only take me a second, actually. Sorry about that. Yeah, thanks for reminding me about that. If I show up, or anybody shows up and it's not posted, it should be, and it only takes me a second to get it. Final signature, what happened. And here we go. One more minute. Well, maybe the upload failed and I just didn't bother to check. Okay, it's there now. I want to pull it up in a second. All right, should have double checked it. Okay, there you go. So now we're getting down to really the extraordinary adaptation in the nervous system of converting an electrical signal, in the vast majority of cases, to a chemical signal, and then back into electrical signal. So the synapse, there's a gap there, and the synapse could be excitatory, inhibitory. Okay, so that gives you an ability to synthesize different inputs, different information, and have that be synthesized, and then the signal going forward would then be somehow a composite of that. All right, so you get a new signal coming out from different signals coming in. And the other thing about the synapse is it's highly dynamic. It is not basically like a node, right, or a transistor. It's not an up or down, okay? To a large degree it is, but it actually has a fair dynamic range. Okay. So we'll talk about release sites, about vesicles, active zones. Some of this will be reviewed from some of what you've done in biology before. We'll get into some classic and really simple experiments. When you look at them, you think, well, that was easy. Why didn't they think of doing that before? And then they were quite telling to really determine that calcium was truly is the trigger for neurotransmitter release at the synapse to release that chemical message. And then the concept of the quantum talk a little bit about synaptic proteins. I think you've gone over this a little bit in CMB, those you haven't. And try to keep it simple. Try to make an effort this year to make it clear, like what you should really focus on. And then in so doing, we're talking about the very basic elements in terms of synaptic release of neurotransmitter, that is the synaptic vesicles responding to calcium entry and fusing with the membrane at the presynaptic nerve ending at what we call the top of the cleft, okay, to dump the chemicals out. So there are stages to that where the vesicles dock, they then become primed, and then they fuse, and we'll talk at the end about the synaptic vesicle cycle. So you can't see it very well down here. It's an electron micrograph of a synapse. It has some classic features. I know some of it would be hard to see, but so this is the presynaptic nerve ending, and you see how this is the axon here, and it's swollen out a little bit, and you can see the vesicles, and these were observed when the the electron microscope itself was brought into use. It was created and brought into use. Some among the first tissues were looked at were brain. You could see these things, see these vesicles, and for a long time people thought, well, you can see vesicles in other cells. Sometimes you can actually see them in the axon. You can see them in non-nerve cells. So it's like, well, they're some kind of storage thing. We don't know what they are. The person who discovered and came up with the idea of the quantum, he discovered the basis of it and came up with the idea of quantum subversion of catch. When he first saw that, he said, there's my quantum. So he realized that this is where the signaling originates. But it took a long time to prove that, and there were a lot of naysayers. At any rate, you can see that the synaptic cleft is very, very, very small. It's about the width of the vesicle. It's very, very tight. Okay, and there's actually material in that. You can, when you do electron microscopy, you're counterstaining, but you're really seeing density. And so you can see there's a lot of dense stuff here, and you can sort of see the vesicles there, but they are all over the place. So that was always a part of it. But it looked like there's what was called we now call an active zone, a site that seems to have a bit of protein on one side, and then in certain synapses you see a pretty heavy density on the other side. We call it postsynaptic density, and that would usually signify this is an excitatory axon. And then here's a little bit of a swelling here, and this would be a spine. So that was another clue now that we know quite well that this would be an excitatory synapse. So all these features are fairly clear, and you can see that on the average, the vesicles, if you look at this closely, are fairly uniform. They're not perfect. Right? They're not exact, all right, but they're fairly uniform. In some nerve endings, they're not uniform at all. There's also often some larger structures. It's not shown here. Usually there's a mitochondria, and usually there's some very, very large, dense core granules. And so this is the picture then that's evolved. So we've seen a variety of different types of synapses. So this looks more classical, like what I just went over in electron micrograph. You have vesicles, but some of them are well away from the cleft. Okay, this again is not showing for simplicity the mitochondria, but we have this density down near the site of the cleft. Remember, the synapse is the whole thing. It's called the clasp. Or that's the derivative from the Greek. But nonetheless, you can see this, what looked like densities. People thought these must be proteins. There must be some machinery there to make this work. Okay, maybe they're channels, maybe it's all the above. We now know that, and didn't see it in the electron micrograph, that almost every synapse has a couple of very large, these are called large dense core vesicles. They could also be called granule-like. There was some question about whether they should be called that, because if you look at a secretory cell down here, whether this is in the pancreas or or wherever, you can take your pick. Okay, maybe even in the adrenal gland. All right, you can see these very, very large granules, and they're bigger than these large dense core vesicles. But it's clear that these were full of proteinaceous-like material or something dense that the best guess is. And there's usually a couple of them. Just a handful, maybe, a few. All right, vast majority of these enclosed structures in here are vesicles. Then you can see some tubular elements, which are a little bit of endoplasmic reticulum, and there may be a few other things like caveoli. But this is what dominates. So for a long time, it's quite clear that there were two structures in the nerve ending. So to be absolutely clear, presynaptic nerve ending, vesicles here, defining that presynaptic nerve ending, okay, and then the postsynaptic part, all right? Now, this would be a classic synapse that you see in the nervous system, so it will be a nerve cell on a nerve cell. There are many examples, though, particularly in the, obviously all through the periphery. Basically, all your organs are innervated, by and large, by autonomic nerves, okay? So they will have a mixture of, again, of these small vesicles, but they'll have a lot of these large dense core vesicles, okay? And so this could be an autonomic synapse. This could be on a blood vessel, like capillary. Could be on some other smooth muscle, say in the bladder or the GI tract, or in the bronchi or wherever. Okay. So they vary. They vary all over the place, all right? Some cases, the alignment of the vesicles is, really quite discrete down near the synapse. In some cases, you can hardly see a vesicle near the synaptic cleft, and most of them are way back. Okay. Now, so the question very early on was, are these vesicles, are they part of the signaling, So one of the first things was done is to isolate vesicles, and you can see neurotransmitter in the vesicles. So okay, yeah, so they are. And somebody said, well, no, they're storage. The synapse works too quickly. You can't have this vesicle come down and fuse. That just takes too long. I've heard people, even when I was a graduate student, heard people saying that kind of thing. So there was a group that did a very cool experiment, and they took synapses, it doesn't matter where it was from, but in this case from the hypothalamus. Okay, they also did it at the frog neuromuscular junction, so this is a nerve muscle preparation. Okay, and they used this machine. So this machine, okay, was cool because what they did is they set up the tissue on this piston, on this head, okay, that they could drive down onto a cooling block. In this case, they were very serious. They used liquid helium, which is about 4 Kelvin, okay? And they also could stimulate the tissue. They set up actually could stimulate the nerve the way it's arranged. So they stimulated it as this was driving down so they could capture, instantly what happens. It went and looked at an electron microscope, and they could see these kinds of things. They called them omega figures. You could start seeing them, and in a couple cases, they could capture a situation where there was a little bit of dense material in the vesicle, and you could see that it was just opened out to the synapse, and it looked as if the material hadn't moved yet. And they could then correlate the degree of stimulation. They could measure how much synaptic release there was of neurotransmitter, and they could see that as you stimulate, you see more and more and more of these things. But it was a correlation. So to get back to some of the features then of the synapse, from some of these experiments and many others, it was clear that there were particular sites where you could get these fusion, or they called them omega figures because it looked like omega, but you could see these little fusion points, okay? And in a very, very well understood, very well defined system, the frog neuromuscular junction, it was first shown that there was a site for docking, and now we know on any muscle, so it's showing a guy with a bicep right there. So you see presynaptic input with the nerve endings here on the muscle, and you blow that up. You can see in there that the synaptic vesicles, when we looked at that from a micrograph, you're taking a real thin slice. So now you look at the whole thing and reconstruct what it looks like, and the vesicles line up. They line up actually, by and large, in contact with muscle in 2 rows. Okay, so there's 2 rows, and then there's sets of 2 rows that are lined up right at that synaptic, or what we call effector junction, the neuromuscular junction, okay? And they're perfectly lined up over where the receptors are. So curiously in muscle, there's these infoldings, these junctional folds. And so it turns out that the receptors are really rich on those folds, and they're the richest right under where you see those rows of docked vesicles. So this now shows there's organization to bring what we now know is where the release happens in neurotransmitter, right opposed to the detector, the receptor. Okay. Why do you think we have some folds in our membranes here? Well, you can see that you have more receptors. If you unfold it, more surface, you have more receptors. You don't see this at all synapses. In fact, it's not clear that you really see that in any synapse in the brain, for example. Okay. So we now know that these vesicles are where the neurotransmitter is. Okay. And so the one thing to point out is that this docking site on the presynaptic side is known as the active zone, separate from the postsynaptic density, where we now know that's where the receptors are. So the active zone, that's just really a formal description of the machinery that's there for the vesicles to dock and do their business. So I'm going to go through these next two things, well, at least this one fairly quickly. So this seems like a ridiculously simple experiment. This was done by Sir Bernard Katz, who really was an extraordinary neuroscientist who really picked out the mechanisms, the fundamental mechanisms of how the synapse works. And again, using a very simple preparation. Used the frog neuromuscular junction, because it was easy to have frogs, and he was in England. He had plenty of frogs there that he could get his hands on, and it was very clear, well-defined preparation. And so he worked out conditions to get very high resolution recording, and that's what's shown here. So if you take the top, what's being done here is actually recording now from the muscle. So you're recording right as close as you can to the synapse. So this will say, okay, if we're going from electrical to chemical back to electrical, which then will contract the muscle, let's capture that second electrical signal, that potential change. And it worked out, quite a wizard at doing this, and worked out the conditions to create microelectrodes to record discrete signals right at the synapse. And so you could use another mechanism, usually through the same pipette, to do a quick stimulation. That's the S, okay? And then right at this position, you record right next to the synapse, and you get this large potential change that would decay. This is not an action potential. This is due to the neurotransmitter release causing a potential change. You actually put a poison in there to prevent the muscle from having, because a muscle can have an action potential, these fibers have it. Action potentials, it's a little more complicated. You might remember the sarcoplasmic reticulum and its role in terms of releasing calcium. All that's blocked. So now you're just seeing the potential that's happening at the nerve ending. And so it goes up and it comes down, right? So, there's a bit of capacitance effect there, the voltage effect, some sodium channels, potassium channels are coming in. But this was not enough to make an action potential. So this is really kind of that rising phase that's decaying. But this is the direct signal from the synapse. A very simple experiment, just move the electrode down away from the synapse. Well, it was already fairly clear that you needed that synapse, because you wouldn't get a potential if you yanked the nerve off, right? Or if you did other things to block that nerve, it wouldn't work, okay? So, in this case, we're stimulating the nerve, so you're getting release, but there's other ways that you could try to see if you could directly stimulate, say, the nerve ending. Okay, not there, won't work, so okay, that's where it has to be happening. But he saw a couple things. One is he moved away, saw that the potential got smaller and smaller. So this meant that you really needed, of course, an action potential to spread the whole potential across the muscle fiber to get a contraction. That was fairly clear and obvious. But it got smaller, but look at it did. It started to flatten out. So this told you that the signal starts there and then it's spreading out. Okay. Now there's a condition where you can have huge upregulation of the receptors. Okay. Usually that happens with paralysis. And so in that condition, you see that it doesn't decay like this. You see that you get a very, very strong signal across the muscle, and so you'll get a walking response in the muscle, a very heavy overcontraction. So it's very simple. That's where the action is. And then he did this experiment. And the prelude to this was fairly obvious. It was, okay, if you take calcium away, you have sodium outside, you have potassium inside, a little potassium outside, some chloride, but you also have calcium and magnesium. Pretty clear that calcium is probably the trigger. Take sodium away, okay, things don't work, but you take calcium away, you stimulate, and you lose the responses. You just do this over and over until the cows come home, you'll get nothing. Now this is an interesting experiment. Instead of stimulating the nerve, they used a pulse of sodium. So there was no sodium here either. Okay, so that was interesting. So that would prevent any kind of action potential. The effect should really be right down at the synapse where it showed that where all the action is. So we give a puff of sodium here. Okay, so now you look at the case where I'm not showing it. When you have calcium there, you get a response, but this case was a little more tricky. Gave a real short pulse of calcium just before that sodium stimulation and got perfectly good responses. So we call these postsynaptic responses. It's the same before. You're recording on the other side of the synapse, you're recording right near the synaptic site. So you're looking at that postsynaptic potential is what we call it. And these look like what you get if you had calcium around there all the time. So then it got tricky and put calcium just after. I thought, okay, you stimulate, channels are open, now we'll add the calcium after. Do we get a response? And we don't. So this meant that the calcium timing of entry was essential and critical for producing this postsynaptic potential. And then the inference is that that's calcium that's triggering the release of neurotransmitter on the inside. So it seems obvious. So okay, this must be the way it works, because you take it away, it doesn't work, you add it back, it'll work. But they could look at the timing very carefully before and after. This is just one snapshot and show that the calcium had to really be there right before. So there's a critical link between the stimulation and the calcium entry. Okay. Now we're going to talk about the quantum, and this will be really one of the two main things we want to cover for the rest of the lecture. One is that people have seen this kind of thing for a long time and ignored it. But Katz was really very intuitive and very alert. So under the right conditions, when you're recording from this nerve muscle preparation, and you have a good signal to noise, and you turn the signal way up, you can see these little blips. And people saw them for years and thought, well, it's something in the background. It's noise. It's electronic noise. They call it noise. They had no idea what it was. And just ignored it. And we're only interested when you stimulate the nerve, when we get a response, that's synaptic transmission, that's important to study. Kat said, no, this is really interesting, because when you blow them up, look what they look like. They look like these. Look at the scale difference here. So this is just a few millivolts, this is 10 millivolts. So this is the full-fledged postsynaptic potential we just saw a minute ago that you're driving the nerve to get, right? So this is, action potential causing whatever degree of neurotransmitter release, so you get this big response. In this case, it prevented it from causing an action potential in the muscle, but if you didn't do that, it would signal. It would cause a action potential and a contraction. So we got these little bits, and they look a lot like this. So we thought, maybe these are building blocks. And in fact, sometimes, if you look through here, they look like they added up on top of each other. So these are spontaneous. We call them spontaneous miniature, because they're mini compared to these guys. End plate potentials. End plate is another name for the neuromuscular junction. Don't be confused by that. It's an old-fashioned term, but it looks like a plate, so they called it the end plate of the nerve. But it's the synapse. Okay, it's the neuromuscular junction synapse. Okay, so they went on and did something more tricky. Is they said, okay, if this is a building block, and we reduce calcium, but we don't get rid of it, not like that other experiment, but we knock it down, then if this is linked to release, this should get smaller and smaller. In fact, they got to the point where they could see that they were just units of each other. They were adding up. And so this first publication with Paul Fatt, so this is one of many Fat Cats publications, we were able to determine that the smallest unit, okay, so this is the size here versus the number. I know this is a little hard to look at. This is what's called Poisson distribution, but you're looking at the size on this axis. Okay, so the littlest one you plot here, and how many did you get? You got a lot of them. This was done under conditions where you lower the calcium, where actually, when you stimulate, most of the time it's not working. So there's actually a whole lot of zero here. And this is the smallest one, and it lined up beautifully with the distribution of the discrete spontaneous one. So I thought, this is the elemental unit. Parallel to physics, we'll call it the quantum. So the smallest one miniature end plate potential, that's the EPP, is the size of a spontaneous end plate potential, and he named that the quantum. This was followed up very shortly thereafter by Martin Boyd's work. Okay, this was Bob Martin, University of Colorado. They actually were able to use a different preparation, tweak it so they could really optimize it, and they could not only see the unit, but they could see beautifully the discrete steps all the way out to, say, 6 summed quantum events. So the idea is that this is a basic unit now, and the hypothesis was that the big thing that you got was built up from all of them. There was a lot of other work to show that that's indeed true. Right? And we now know that this is the building block that was synaptic transmission, that it's each vesicle fusing, and then as they add together, that produces the extent of neurotransmitter you get into the cleft. So if it's 1 or 1,000, obviously if it's 1,000, that's a lot of transmitter. If it's 1, it's very little, and that causes a potential change, which is in direct proportion to that neurotransmitter coming out. More neurotransmitter, more activation on the postsynaptic side, bigger potential, right? Okay. So to really nail it down, though, the question was, okay, if it really is due to release of neurotransmitter from that packet, can we mimic that? And this is where Stephen Cooper came in, another extraordinary neuroscientist who was extremely creative. So I'm not going to go through this in great detail, but just enough so you understand it. And so what he wanted to do is, can we create a vesicle? That's the bottom line. So he used a preparation, basically the same preparation, the neuromuscular junction, okay? And in the beginning, they took off the nerve, but then later they got so good at it, they could leave the nerve on there. And why did they do that? They could compare what they would get with their artificial vesicle to a spontaneous vesicle. And that's exactly what's shown here. So here's the trick. You basically have a tube, all right? In this case, the neurotransmitter that drives the system, that's acetylcholine. So you have a little tube of acetylcholine. You know the concentration. You can take out a certain amount of that, and then they create a vesicle. And they do this in a layer of oil. So you can see it. And it makes a discrete vesicle because it's water. And so now you get a little bubble of water, or in this case, the solution of acetylcholine in the oil. So they would basically pump it out and then very carefully measure the diameter of that thing. Because you knew the concentration, know the diameter, you know the number of molecules then, right? You have your volume, concentration, it pops right out of it. On the right conditions, they pushed it right down on the neuromuscular junction on that synapse, and they could get a response. So that's the applied vesicle, and they caught it beautifully in one case, where actually I have 2 cases here, one by itself, and one where just after that happened, the nerve popped out a spontaneous fusion that is a miniature end plate potential, and it was basically exactly the same size. And they could go back and calculate. There's about 10. They want to know exactly how many molecules were in the vesicle. I don't know if that's a number that means anything. It's not like Planck's constant, right? So they could determine, though, it was just, in the large and short of it, that there was it didn't take that many vesicles to get a potential change, 10,000 molecules. That's not a lot, okay? So a very insightful experiment said, okay, let's create a vesicle. Does that work? And can we get one that looks just like the spontaneous one? And in fact, they did. So that meant then that, by inference, that the quantum, again, is due to one vesicle. So what really made the final connection was, and it took some years later until we got hard numbers, we measured very carefully the number of molecules in one vesicle, the real vesicles from the nerve, the numbers match pretty closely. Okay. I'm going to skip this because of time in terms of going through the detail. All I'm going to say is there's another trick you can use to try to measure whether the membrane fuses, and that is when the membrane fuses, it adds more membrane. Remember, the amount of membrane is what dictates the capacitance. So there's a way to measure capacitance. You can measure the capacitance. In fact, you'll see a jump in capacitance with fusion. So these were some of the most definitive experiments to show that indeed that the vesicle, when it comes down and fuses, it has to do that to release neurotransmitter. In fact, they're able to use a detector to detect the neurotransmitter and a method to measure the fusion. So you can see the change in capacitance of the fusion, and then the neurotransmitter. So from both sides, you can see that you have the vesicle came down. It accounts for the quantum, but it has to fuse. Okay, it's not somehow dumping its contents out and they're getting out somehow. Okay, so if you want to know a little more about the details of this, we can go through that later. And the last thing I want to go over, I know we don't have a lot of time, but I want to really focus this down, and I think a lot of this is familiar already. So I think those of you who had CMB, or most of you who had equivalent, that you know about snares. So you know about membrane elements within the cell. Usually it's just talking about this with the endoplasmic reticulum and then movement of proteins through the Golgi, that things have to go from one membrane compartment to another through vesicles. So you have fusion inside the cell. Discover that this involves these things called snares. Well, they actually are all out at the nerve ending. So initially there are these snares, a set on the vesicle itself and a set on the target postsynaptic membrane that have to come together, and that's the initial docking event. So the vesicle, you know, we saw that they're all around the inside of the presynaptic terminal, but there are a number of them that are docked. Okay, so the reason the system is fast is that there are a bunch of vesicles that are down there docked, ready to go, but it's not quite enough. So the machinery actually has to undergo what we call priming. So it's not only docked, but it's just about ready to go. So you can see the difference between docking, and this is an approximation, but it is a fair approximation, that with docking, the vesicle isn't really necessarily all that close yet to the membrane, but it's ratcheted down right next to the release site, to the release membrane, with this priming, where these structures start to really wind together. Okay. So there are a whole lot of proteins involved with synaptic transmission, particularly with vesicle fusion as well. And of course, there's calcium channels there, right? So I've highlighted the ones that are important to know. So with docking, we have a pair that are essential, but we see them replicated down here in fusion. So something called synaptobrevin, and it is termed a V or vesicle snare. So it's on the synaptic vesicle, was identified there in just basic biochemistry that was done on vesicles, purified vesicles, what's there. The major protein that was found was the synaptobrevin. It's also called VAMP. Okay. And then there were two other ones that are found on the target. So they were from what we call synaptic membranes, but we really mean the presynaptic membrane, the target for the vesicle. Okay. And those are SNAP25 and syntaxin. So key thing that's important as well about the syntaxin to know is that this is a target snare that interacts and coordinates with the calcium channels. So then so there's a lot of other proteins here, and you can read about them, and they're interesting, but the only other one that counts is the synaptotagmin, and that's the calcium sensor. Okay. So if you look at what happens through these basic events, this is another picture of that, all right, that we get this docking initially, so interaction with the synaptobrevin with the SNAP25 and syntaxin, so they're on the target membrane and the presynaptic side, okay? So they dock, and then they go through this priming event. So the V snare and the T snare, that's synaptobrevin, and the T snares, they interact, okay? So they start to interact, and it's interesting. You see they're elongated structures. They have large alpha helices, so they got these rods. And the rods kind of come together, okay? And then they turn to the side. You see how they're off to the side? And that's what's ratcheting down the vesicle. So if there's anything from this that's essential for you to understand, is it's not just that there's proteins and they bring things together, but the way they structurally interact, they are bringing down the membrane so close that in fact they're ready to fuse. And guess what? Without a signal, it happens, right? And why do you know it happens? From Katz's experiments. Remember there's spontaneous neurotransmitter release? So sometimes this just goes by itself. So it's really, really ready to go. So that's a big part of the fact that it's primed to signal, even though the vesicles are all over the place. So it turns out when one set of vesicles fuse, new ones come right in, they prime. So the system's ready to go. It's not like the vesicles have to come down with the calcium. So you get axe potential, calcium comes in. Vesicles aren't migrating down and then fusing. Too long. Okay, they're ready to go, and they're ready to go because they're ready and sitting there all basically ratcheted down, and then there's this, so they zipper up, there's this synaptotagmin, and what is it doing? See, it's the calcium sensor, but it turns out it's the brake. So it sits on the snare complex, the V and T snare complex, and holds it from going all the way to cause fusion of the membrane. So when calcium comes in, the synaptotagmin, okay, there's steps inside here. Synaptotagmin is released. This fully ratchets down, the membranes fuse, and so the vesicle fuses with the target membrane, contents come out, and then there's some proteins that we're going to not worry about that have to come and sort of break this whole system back down so it can start the cycle again. And that's the synaptic exocytosis or fusion cycle. Okay. So who's essential and why are we, there's all these other proteins, right? Well, if you just take synaptobrevin, the SNAP25 and syntaxin, okay, and you stick them in so you put the synaptobrevin in one cell, just a regular cell, not a nerve cell, like what we call a fibroblast or endothelial cell. And you put the target ones in another cell. So you put the SNAP25 and syntaxin, and you let them bounce together. They will actually dock and fuse. Okay, so inherent, they're really slow at doing that, but inherent in these structures and that interaction is all that's needed for the machinery to come together and make what we call a fusion pore. Okay? So key to know are these basic elements here, all right? And then, of course, when the recycling will involve to varying degrees endocytosis, because you got to make a new vesicle again. Okay. So this is just shown as a cool picture where you, again, you have the synaptobrevin, the SNAP25, and the syntaxin, and just reinforcing that they're these rod-like alpha helices, and they coil around each other. And how tight those coils happen, that's the zippering. And the tighter they get, the more they bring the vesicle in cross-section here down to the presynaptic membrane. And then there has to be some kind of the lipid fusion event. Okay. Now this is something just to look at. All right. I don't want you to know these guys, but the whole point is that there's actually a collective of proteins that come together. And probably the key thing right here is to show that the calcium channel is interacting with the syntaxin in this large complex, so that all this happens together, all right, where the calcium channel and the assembly of the fusion complex, okay, they're all coordinated together in this active zone. So the calcium entry is right where the vesicle is, okay, through this coordinate interaction of all these other proteins I don't want you to know. Okay, it's tethered, there's all this interaction to bring this all close together here. So when this thing, the snare complex, eventually is released, get full tight interaction to bring down the vesicle to fuse. Okay, that's all coordinated here. I'm going to skip this. This is just an idea that has been proposed for how you actually get the lipids to cooperate. Okay, and the last thing is, okay, well, this goes back to something that I showed in a very early lecture. Actually, it was in the overview. Skipped over this. So, well, what really does this look like when you're pairing the incoming signal, the incoming digital signal, which is the action potential, with the outgoing signal, which should be the action potential, right? If it's excitatory. So this is showing then that when the action potential comes in, this isn't the squid giant synapse, which connects to the squid giant axon. Here's an action potential coming in. So this is recording on the presynaptic side. Okay, and then you can see as recorded on the postsynaptic side, there's the postsynaptic excitatory postsynaptic potential. That's the release of neurotransmitter. Okay, so you can see that if you add TTX in on the other side, or you have a way of blocking the action potential on the other side. Take your pick, whatever way you want to do it. Okay, tricky to do, but you can. So that would be the underlying thing. Now it's at threshold, so it will trigger an action potential in the postsynaptic side. So the key thing is that you see that the main release of neurotransmitter is happening at the back end of the action potential. Okay? So we would say the falling phase of the action potential. And there's a little question there as to why. And the last thing is just to show the whole thing as it cycles from docking to priming to fusion and going back through this. And this is what's happening in your synapses all the time, every day, so that you can not only generate signals, but you can regenerate. I'll let you references at the end. Okay. Well, we ran all the way up to the end of the tank.